it's the tool for developing and screening drugs. So this is something that we can do. Imagine you have a drug, you want to see what it do on cells. The first thing you want to do is to add that drug to the cell. Is it going to kill them? Is it going to increase their proliferation? Is it going to increase certain proteins, decrease certain proteins, or change the function? We can also use cell culture to study carcinogenesis and mutagenesis. So imagine you are introducing a mutation to a protein. Then you want to know how the function of that protein is affected by that mutation. Then cell lines are the best thing to use. You can study the physiology and the biochemistry at the resting state or the natural state, as well as the diseased state. So imagine you have somebody with myocardial infarction, for example, in the heart. So the cells that come from that heart, they are diseased. So if you take those cells and you culture them and you look at their biochemistry, they are totally different from the ones from a normal heart. And finally, we can check the biohazards and the drug efficacy. So here you are checking the development and the screening, and also you can check the efficacy. And there is multiple models that you can test in cell cultures, starting from using them as a model system up to using them to tissue bioengineering aspects, because you can have those cells. And now we have these days cells that are mounted on membranes, and those films can be grafted back, for example, in organs. So all these aspects, like the toxicity testing, the viral production, in terms of cancer research, in terms of genetic engineering and manipulation, in gene therapy and stem cell therapy, up to the tissue bioengineering, all that can be very useful to have the cell lines and cell culture available. Of course, this is a very long field that has been studied since ages. So this is the history of how the cell culture started. And there are some major building blocks that happens throughout the years up till 1980s, where the tissue culture start to be really one of the fundamentals of the research, and that is widely used and accepted nowadays. So how can we identify cells based on what, and what are the types of cells that normally we use in cell culture? So as you know, the cells, they have, if you look under the microscope, the first thing you will see, the cells are characterized by their morphology. So you can tell quickly that this is a fibroblast because it's an elongated cell. And you can tell that this is an epithelial cell because it's a polygon-like attached to a plate. It becomes more difficult when you go to specialized cells like endothelial and neural cells, because their culture is like a little bit more difficult. There would be some more requirement in terms of the environment. But broadly, you can divide the cell culture into three different types. The epithelial-like cell, and those are the cells that attach very nicely to a plate, and these appear flattened and polygonal shape. So I wish there will be a lab associated to this course, because I could have shown you in the next session those under a microscope. But unfortunately, this is not the case. Then the other type of cells, this is the lymphoblast-like, and what differentiated these cells from epithelial cells is that they don't attach, and these remain in suspension. So if you grow these cells, they will be in a flask and they will not attach to the surface. And finally, there is the fibroblast-like cells, these would attach to the substrate, but they are elongated bipolar, so they are totally different from the epithelial cells. Now, it's important for you to know that fibroblasts are cells that do not have too many markers. So epithelial cells, they have a marker, endothelial cells, they have a marker, neuronal cells, they have a marker, so you can identify those three types of cells very easily. But fibroblasts, you cannot. Okay. Could you please remind me what time we end the lecture?
12:50.
12:50. Okay. So this is an example for showing you the importance of how to understand the cell culture from a perspective where the cell came from. So if we look at an epithelial cell, you see they are really very well-defined cells. And normally they are making the lining of, for example, the intestine and other organs. And these are attached to a basement membrane. So the first attachment of these cells with the basement membrane is mediated by certain specialized protein like integrins. Now, if you look at this basement membrane or the extracellular matrix, there's a lot of high molecular weight proteins like collagens, laminins, and proteoglycans. So you have to think, like, if you want to get those cells out from a tissue, like say you take this tissue and you digested it, you put some enzymes so the cells are dismantled, and then you want to grow the epithelial cells. You have to think where they were lying before, on which membrane, so that you can prepare them to be cultured in a culture dish. So these things, we take it into consideration when we do the cultures. Because if you take the epithelial cells, they are lying on this basement membrane. So this component is going to be useful to grow epithelial cells. If you look lower, we have the endothelial cells, which are the lumen cells of the vessels. Again, they share one component, which is the integrin, but you can see that the epithelial cells, the endothelial cells, they are much smaller and they are much thinner. And most probably, the basement membrane here is a little bit different from the basement membrane there. So the growth requirement of an epithelial cell is different from an endothelial cell. And this is translated later in real culture condition, where we add different material for epithelial cells to grow in the growth medium compared to the endothelial cells. So the number one rule when you get to any lab where you want to grow cells in culture is to know what is the cell type, from where it comes, and what is the right growth medium to use. Okay? Now, the third cell type you see here is the fibroblast, which look like it's in the tissue away from the basement membranes. So this cell, it's able to grow in any connective tissue, and therefore it would be the easiest to grow in culture. That's why fibroblasts are so easy to grow in culture. If you take them here, they don't really need a very specialized membrane to grow. And therefore, if you dismantle this tissue, a fibroblast would be very easy to grow anywhere. Okay, so how do we prepare a cell culture? To start with, any cell culture comes from a tissue. Meaning, if we use mice as an example, let's say you want to make skin tissue, you need to go take the skin from that mouse and use the right components of enzymes in order to dismantle the tissue. So remove the basement membranes and cut it in into small parts. And after that, you will give those recessed tissue the right growth medium, and you add it into plates. And that would be the first culture you're doing. So for example, I'm gonna add for you a couple of videos after this lecture to show you how I prepare fibroblasts from the heart. So I take the heart and I digest it with some enzymes like trypsin and collagenase. So the cells are dismantled. And then I will put the cells in the growth media, and you will see after 96 hours, I will get to the primary culture. And then I will do another subculture when the cells become filling the plate into a secondary culture. Okay, now if you want to get a cell line is defined to be a cell that is immortalized. Meaning this cell line, you have a cell that is able to divide for a long, long period of time. But let's go first to discuss what happens in the primary culture. What do we call a primary culture? Because when you recess a tissue, you have multiple cells there. So initially, this is a heterogeneous culture. So there are many cells, many cell types, endothelial cells, fibroblasts, epithelial cells, and it has a finite lifespan. So this means it cannot grow more than 3 or 4 times. And it retained differentiated phenotype, because if you go back here, this cell and this cell, they are totally different. Endothelial and epithelial are way different because this is differentiation. This cell has multiple forms of differentiation to become epithelial cells, while endothelial cells also has different types of differentiation. So if you get this culture, you will retain differentiated phenotype. In other words, you will have epithelial and endothelial and fibroblast mixed together. And it exhibits contact inhibition. So at this point, when they become confluent, if the cells contact each other so much, they will stop growing. And they need a support to grow. Again, if you look back, this cell needs a support, this cell needs a different support, and this cell has another type of support in the connective tissue. So you maintain more or less what the tissue looked like in the primary culture. So it's anchorage dependent. When you subculture this, it will look like mostly like the primary culture, but most probably you're gonna lose some sort of cells, some types of cells, like isolated by selection or cloning, and it's more homogeneous population. Because when you do the subculture, most probably some of the cells die because the growth medium you are using is gonna not be useful for all the cells. Some of them die and some of them are retained. Now, to maintain the cell line, you have to immortalize it. And in the next slide, I will describe to you how we do a cell line immortalization. But let's say now you took the heart, you took the cell, and you subculture it. You are at this point. Either it will stop dividing because that's the capacity, 3 or 4 passages, that's it, because it goes for senescence, or you will select a single cell from that and you will do clonal cell line, and I will describe this in the next slide. Or you simply take all those cells and immortalize them by one of the two ways. One way is to keep growing the cells and subculturing them, or you transform them by certain proteins that comes in the next slide. So as an example, transformed cell, immortalized cell lines are most of the cancer cell lines that we use in the lab, because these cells grow very fast and they are already uh, uh, have a different gene transcription profile. So just if in case you don't have a good background about cell differentiation and regeneration, I just posted this figure for you here to let you know the difference between totipotent, pluripotent, and differentiated cells. So any of the cells that we described here, epithelium, endothelium, and fibroblasts, these are differentiated cells. But the magnitude of differentiation is different, meaning epithelial cells, they can only generate epithelial cells and probably some other cells under stress conditions. Endothelial cells will be always endothelial cells. Fibroblasts probably can differentiate to something else. So the capacity to change from one cell type to another after differentiation is very difficult. And therefore, the more you move from the totipotency of the stem cells, the more you are going to the differentiation or the specialized cell types. Because when you say differentiated, this means the cell is specialized to perform a certain function. As an epithelial cell is in a lining, so it will absorb certain solutes and certain vitamins, and it will block the entry of others. So that's, for example, how epithelial cell work. The endothelial cell work, of course, by making the lining of the vessels, and it plays an important role in the inflammatory process and all that. So the more you go toward differentiation, the less you are in terms of the regeneration competency. So stem cells are super regenerative, and they can generate many different types of cells. However, specialized cells, they can perform a very specialized function, but they cannot regenerate other or transform into different cell types. So this is also important to know, because most of the cancer cell lines that you use in culture, these will be differentiated ones. So what are the advantages and what are the disadvantages of the cell culture? The advantages are, this is a continuous thing that you can have, and especially when you have immortalized cells, you can grow them in culture indefinitely. The advantage of having a cell culture is that they could be achieved spontaneously. So as I was describing for you here, if you take this cell and you keep growing it, like growing it and passing it again and again and again, you will end up having an immortalized cell line just because you passed it like 30 or 40 times. These can be achieved by transformation vectors, and I will describe the transformation vectors in the next slide. They have a very fast rate of growth, because if you want to do an experiment in a lab, you don't want to do it in like 4 or 5 weeks or 1 year. The growth rate of these cells is really quick, so sometimes you split them 2 times per week in a culture dish. So they also present loss of anchorage dependency and contact inhibition. So because with the way we culture them, you are able to grow them on culture dishes, and you are able to see their confluency, and you can separate them before they become very confluent and their inhibition is their growth is inhibited by contact. And they have an infinite lifespan, meaning you can grow them up to 40, 50 passages. Now, it's important to know that it's not recommended to use them long time. We prepare them and we keep them frozen so we can come back from time to time to use. What are the limitations? The more you keep them in culture, the more they will become genetically unstable because they will acquire more mutations. And they are definitely smaller in size and more rounded compared to what you see in a tissue, and they have a higher nucleus to cytoplasm ratio. So the nucleus is much bigger compared to what you see in a tissue. Of course, their phenotype will differ from the tissue you bring, like where you isolated these cells from. And after a while, they will stop tissue-specific gene expression. And that could be a problem because this is a loss of differentiation. I want to stop here and ask you if you have any questions. Any questions, any comments? Am I going fast? All good? No comment? Okay. So I think I will stop after this slide and we continue probably in the next week. So I will describe this slide here and I'll give you a break for today. So how do we achieve to immortalize cells? So there are several methods to immortalize mammalian cells. First, by using viral genes. And why do we like viral genes? Just to let you know, whenever a virus is infecting our cells, most probably parts of the viral genome is inserted to our genome. Particularly, we are speaking about the simian virus 40, SV40. This you will hear a lot if you go to any lab to work. And the expression of the protein called T antigen of simian virus 40, it causes the cells to be immortalized. So therefore, if you have a cell line like this, you already got the primary culture, you get the cells, you grow them, they become confluent, they fill all the plate, they grow them again by subculture, and then you grow them to step 4. And then if you come here and you infect them with a virus that gives them an overexpression of the large T antigen from the simian virus SV40, then these cells will be transformed. They will become immortalized. 2 weeks after that, most probably the cells will be 100% immortalized and they will become like a cell line. Okay, there are, of course, many other methods. So one of the things that simian virus SV40 T antigen does is that it will induce telomerase activity in infected cells. You know, the cells, they stop dividing because they lose their telomeres. And the other protein that is known to do that is the telomerase reverse transcriptase protein, the TERT. This is another protein. If you overexpress it in cells, you will maintain the telomerases, and therefore you can transform the cells into a cell line. Other things that can be achieved by SV40, it would be by inactivating tumor suppressor genes because these block the growth, like P53 and RB. And finally, we can overexpress oncogenes like RAS and MYC, and those can cause the cells to be immortalized. Okay. I would like to stop here, but before leaving, I would like to really go back and give you a few time to tell me if you have anything that you didn't get, or if there's anything that I went very quickly. It's important for me because this lecture is going to be like a model for the next lectures. So if I'm going quick, I should slow down. If there is information to be added to the slides, let me know. If particularly I set something that you need a reference that is not on the slide, also I would like to know. So it's your chance today to put the train on the right track, if this is the right expression.
Professor,
Mhm.
for the slide on the historical perspective of cell cultures, we went over
Mhm.
that really quickly. Am I to assume like we don't need to know like the specifics of who did what and when?
Exactly.
Okay,
So
thank you.
uh, if I don't really stop much on the slide when I'm describing, this is just to give you a perspective that if someone would like to really know more, you guys can go and read more. But if I don't really say that this is important, also this is not going to be important. Many times for me, when I prepare the lectures, I would like to go like in a logical manner, and not necessarily that all the material are going to be used for examination or assignments. Any other questions?
I also have a question, Professor.
Yeah.
On the slide where we're talking about a method for immortalization, immortalization, I'm not sure
Mm-hmm.
if I understand what it means. Like this slide just shows one method of how to do this.
Mm-hmm. The immortalization process?
Yes.
Yeah. So the immortalization process is simply by overexpressing